Our next speaker is Nicola Neretti, Professor of Molecular Biology, Cell Biology, and Biochemistry at Brown.
Hi, thank you everybody for coming to attend this great meeting. I've been learning amazing things today. My talk is about mostly molecular biology, and I think it started this morning with aging, and I'm going to close with aging as well. So first, the motivation. Why do we work on aging? And well, we know that we have a problem in terms of the expansion of the elderly population. People over the age of 65 is going to increase dramatically in the next year. And there is already a prediction that the healthcare cost is going to be higher than the GDP in the next few years as well. And that's partly due to the fact that there is an increase in the cost in treating chronic diseases. So the motivation is that aging is the single most important factor, risk factor in many chronic illnesses, as we know. Diabetes, heart disease, cancer, neurodegenerative diseases, we've heard about it today. So the rationale of working and trying to ameliorate aging is that if we can come up with interventions that can slow aging, we can, they have the potential to prevent or at least retard the onset of these debilitating diseases. I agree with what George Hurst said this morning. Is the goal is not to live up to 150. It's actually to live as much as we can, but in health, in good health. You don't want to have any of these diseases or many of them together, which is this comorbidity that happens with age. And so over the course of the many years that people have talked about this problem and tested it in model systems, we have identified a lot of hallmarks of aging. And as you can see here, there's many of them. In general, they go from mitochondrial dysfunction, so energetic problems, stem cell exhaustions, cellular communication gets impaired with aging. There's genomic instability. There's a telomere shortening. A lot of you probably have heard about that. Epigenomic alterations. Loss of proteostasis and cellular senescence, and deregulation of nutrient sensing. So that, again, energetic problems. And my lab has been involved primarily in the study of three of these hallmarks, which is cellular senescence, genomic instability, and epigenomic alterations. So what will come most of my talk is about cellular senescence, so I want to introduce cellular senescence for the people who are not familiar with it. So this is the cell cycle. The cell spends most of its time in G1, just hanging around, and then the DNA is replicated in S until the cell is actually divided through mitosis, and you get two cells. So dividing cells in our body go through this cycle all the time. And what happens in cellular senescence is when the cell senses that there is a certain degree of damage to any of the biomolecules, it could, particularly DNA, the cell exits the cell cycle and becomes senescence. What happens when that occurs? Well, so senescent cells have both positive and negative consequences. They have a functional role, especially in wound healing, and also in, as a means to prevent the onset of cancer. So if you have DNA damage, which could be an oncogenic insult to the cell, a cell tends to become senescent to avoid overproliferation. But at the same time, people realize that these senescent cells, because they survive even without dividing, the body has to clear them. And the way it is cleared is senescent cells secrete what is called a senescence associated secretory phenotype. It's an inflammatory signal composed of many cytokines like IL-6, IL-1, and many others, which signals the immune system that something is going wrong. The immune system comes in and clears the senescent cells. So this is the typical pathway that is used by senescent cells that signaling that something is going wrong. But the problem is that over the course of time, as we age, the immune system efficiency lowers, and this leads to an accumulation of senescent cells with age. And we have seen this in tissues. What does that mean? It means that you have more of these cells and more of this signaling that goes on in your tissues. And what does that lead to? That leads to increase of age-associated chronic inflammation that nowadays is considered one of the hallmarks of aging and probably is a co-causal to many diseases, or at least has a component in many of the age-associated diseases. So how do we know that this is actually important? I want to show you two mice from an experiment a few years ago. This is not from my lab. This is from Baker et al. That was published. It was one of the first real clear evidence of what's going on. So which one is the older of the two? It's pretty clear. These are like wrinkly ears, and the fur is not good. The eyes are not good as well. These look much, much younger. But as you can probably understand, I show you this picture because these are the same exact age. They are 22 months old mice. The difference is that in these mice were engineered with an intact gene which senses the activation of one of the genes that gets activated when cells become senescent. So it is possible to essentially induce all of senescent cells in this mouse to be killed, to go into apoptosis and clear them. So same genotype, just apart from this means of clearing senescent cells, 22 months old, and this is, younger looking, and also in terms of pathologies, this has much fewer pathologies. And when they looked at the survivorship, this is survival for the two genotypes here, there was a 27% increase in survivorship. So they not only live longer, but they also look younger. And this is one of the demonstrations of what I said at the beginning. If we can actually push an organism to live longer, it will also delay the onset of aging problems. So my lab has actually tried to characterize better these senescent cells. The reason is, as you can see, this is a genetically engineered organism. If we want to translate that into human therapeutics, we will have to use a different approach. And typically, the approaches that are used right now involve drugs that can induce these cells into senescence, but they have side effects. They don't only induce senescent cells into apoptosis, but also normal cells into apoptosis, so that's one of the problems. We need a better characterization of the senescent cells to create better drugs to target them. Or the alternative is to actually, and these are called senolytic drugs, or the alternative is to leave the senescent cells where they are, but turn off the inflammatory signal. And those are called senostatic drugs. So these are two approaches that people are trying to develop in terms of drug development in this field. And so better characterization will give us a better design of the drugs. What we want to understand right now, because we have the means, and this is driven by the recent technology that allows us to see differences between cells, individual cells, is we want to better characterize the heterogeneity of these senescent cells. And typically, senescent cells come in different flavors depending on the insult. So replicative senescence is a type of senescence that is induced by shortening of the telomeres. They become very short and the cells cannot replicate anymore. Oncogene-induced senescence, if you turn on an oncogene, these cells start to proliferate very quickly and the replication forks collapses. They get a lot of DNA damage and then they stop into senescence. And also there are other damage-induced senescence that you can induce through agents like etoposide or other drugs that can induce senescence in a variety of cells. And are there other types of senescence? So recently what we have done is we have taken advantage of the recent technology that allows us to study gene expression at the level of individual cells. In the past, we were able to study gene expression, so what is the level of all the genes in the transcriptome, in the genome, only at the level of bulks of cells. You would just extract RNA from thousands of millions of cells and see what happens. But if you want to characterize heterogeneity, we have to look at individual cells. And when we did that, so we did an experiment, this was in collaboration with Tamir Chandra at the University of Edinburgh. We took cells, we induced them into senescence, and we profiled them across several days. Within 7 days, they all become senescent. And then we FACS sorted, and in this experiment, we analyzed the level of expression of every individual gene in all of the cells in the sample. And when we clustered the cells based on the similarity of the gene expression levels across the different times. This is what we saw. So the blue here is the growing cells, and this is green and purple as they become senescence. So each one of these circles is actually one cell in the experiment. And this is a scatter plot where two cells are close to each other. Their gene expression patterns are very similar to each other. And so we saw that the senescent cells were actually clustered in two separate clusters. These, when we looked at the level of expression of the genes, resembled what we know about oncogene-induced senescence. These were different. They were secondary senescent cells. And I don't have time to get into this with more details, but to cut the story short, this secondary senescence is a secondary senescence that involves the Notch pathway. And the Notch pathway, essentially, what it does is a cell becomes senescence, and the contact between these cells of the senescence with the neighboring cells induces the neighboring cells into senescence. And so what this means that this cell is the one that has DNA damage, but it can also influence the cells that are nearby. And so just one cell can influence many other cells. And this was important because there is an idea that SASP signal also can induce senescence in the neighboring cells, but that's secreted. And so if that SASP signal could be the primary inducer of secondary senescence, the signal for senescence would spread into an entire tissue. But we never see an entire tissue becoming senescence. We also see foci. So we think that this mechanism that requires contact might be a way that limits the spread of the senescent phenotype in tissue to a localized area that can possibly be cleared later. Where are these senescent cells? So we actually have a collaboration with Ashley Webb Lab at Brown, and we're starting to look into tissues. And the first tissue we looked at was the brain of aged mice. These are, I believe, 18 months old mice. And we performed single-cell transcriptomics, so a similar experiment to what I presented you. And these are oligodendocite progenitor cells. And again, clustering all of the cells based on the similarity of their expression level. And we found two clusters. And then when we looked at the specific genes that were expressed in the two clusters, we noticed that the P16 genes, which is typically of senescent cells, is really enriched in this cluster here. Which is an indication that we can identify potentially senescent cells in the brain of these mice. And this also happened in microglia from, again, old mice. We had a signal here. We couldn't detect P16, which is a hallmark of senescence, but we could detect some of the signals that involved inflammation and is related to senescence. So here we detected some inflammation in these microglia, especially in the old mice. So this is the story about trying to fish out these cells in the body based on what we know about their transcriptional profile, finding if they're primary or secondary senescence. And the second part of my talk talks about, instead of just looking at gene expression, what are the other features that these cells have that we can use to target them. And this has to do with not gene expression, but with the epigenome. So things that are related to controlling the accessibility of the genome to the expression, the machinery that expresses gene. And so just as a very, very simple primer, because I don't have time to go into the details of the epigenomics and chromatin, but for what I will show you, the important thing to understand is this is the organization of a chromosome. If you could unravel this outside of the nucleus, this is the nucleus. If you could unravel this, it's organized, and I'll talk about the organization in a second. But it is wrapped around these octamers that are called nucleosomes. And there are some regions in which the packing is very tight. It is called heterochromatin, and it is very close. So if you try to go in and read the DNA here, it's more difficult. So they are kind of inactive. And there's regions in which they're more sparse, and they're open, and then the machinery can go in and read the genes and express the genes here. So closed chromatin and open chromatin. And early on, in collaboration with the Sedevi lab at Brown, we started using assays to probe the openness or the closeness of the genome in senescent cells, and this is what we found. So what you can see here, this is called FAIR. It's a technique to probe accessibility. If you look at the proliferating cells, the normal cells that are going through that cell cycle, you see regions that are very accessible, regions that are not accessible, and so on and so forth. And in senescent cells, you get this redistribution. The heterochromatin, the closed region, gets more open. And this region here, as you can see, these are the genes that are in these regions. There's not many genes. This is just a small section of the genome. Typically, in these regions that are closed normally and become open, there's not a lot of genes. So what's in those regions? We realized that early on that the reason why they are closed is because they harbor transposable element. And the first talk this morning, there was a talk about line ones. What are transposable elements? They are actually mobile elements in the genome that can replicate themselves and jump to other places. And you don't want that because they are highly oncogenic, because they can create mutations that can then either impact the function of the cell or lead to other more important problems. And roughly half of our genome is composed of these transposable elements. Most of them are not active anymore, but some of them are, and they reside in those regions that are compact. And so the model that we have, and this is several years of work in the field, is that what happens is these regions that you saw that become open actually usually reside at the periphery of the nucleus, and they're very compact, and they are also touching the periphery, and they're kept closed. And this is where these transposable elements reside. And what happens during senescence, they actually become detached from these regions. They become more open, and they become expressed. And when they become expressed, they can go into the cytoplasm. They get loaded into the machinery to retrotranscribe them. They come in, and they can jump to other regions. And this, in the recent work, again in collaboration with the Sedevi lab, was shown that it can lead to an inflammatory response. The fact that these are in the cytoplasm are recognized by the cell as a virus, because these are actually retroviruses in many cases, and that induces the activation of the cGAS sting pathway and the interferon response. So going back to this whole study, what you have seen now is a one-dimensional view of the genome. You see open, not open, but it's a one-dimensional view. But as I showed you in this cartoon, the genome is actually organized in three-dimensional structure. And so as you can see, the fact that certain regions are more open or more closed has to do with the three-dimensional structure of the genome. And so what we wanted to see is, can we actually measure this three-dimensional structure in senescent cells and see how it changes? And what we did, we took advantage of a technology that was developed a few years ago that is called Hi-C. And essentially, this technology ligates fragments of DNA that are close proximity in the nucleus, but they could be very far in terms of genomic coordinates. And so you create these chimeric reads in which one side of it is one part of the genome, the other side of it comes from a very far away part of the genome in terms of coordinate, but in the cell, in the nucleus, they're very close to each other. By sequencing, with a paradigm sequencing these fragments, it is possible to map them to their origin in the genome and to create a contact map. This is a symmetric contact map where you have all the possible position in the chromosome, and these signal indicates what is the probability of two things being close to each other. And what we know from these studies is that the genome is organized at different levels. We have an organization at the global level in which you have regions that tend to interact with each other. Active regions tend to interact with each other more than inactive regions, which tends to interact with each other as well. And then at the local level, you have these regions of high interaction. They're called TADs. And these are regions that interact a lot because they link genes to their regulatory elements. So a gene tends to be close to another part of the genome that is involved in regulating the activation of that gene. And what we notice is that in terms of these compartments, the active and inactive compartment, we saw a lot of regions that were active in active compartment in proliferating cells, that they became inactive in the senescent cells. Basically, there was a switch in their three-dimensional structure. And also the opposite, some regions that were inactive that became active. And when we looked at the gene expression in those regions, we saw essentially what we expected, which is the regions that go from active to inactive tend to have genes that are turned off, and the regions that went from inactive to active tends to have regions that are turned on. So a lot of the changes in gene expression in senescent cells are actually driven by the three-dimensional changes of the genome. Then we started using imaging. Can we also use imaging and microscopy to see these 3D structures? And that's when we use chromosome paint. With chromosome paint, you can actually target a whole chromosome with fluorescently labeled probes, and you can visualize them. So what you see here is two chromosomes, a chromosome 18 and chromosome 4. And as you can see, this is a quiescent cells or a normal cells. Proliferating are very similar. And this is a senescent cells. And what happens is these chromosomes, in terms of volume, when it is quantified, they shrink in volume. They shrink in volume. So the volume of the senescent cells drops dramatically in both chromosomes, as you can see here, from this to that. So chromosome becomes smaller. But then there's a part of the chromosome that is very heterochromatic and very close, which is the central part of the chromosome. It's called the centromere. So when we label the centromeres, this is what happened. These are the centromeres, this very compact part in a quiescent cell. This is what happens in a senescent cells. They pretty much expand. So this is a visual indication of that loss of condensation of closeness of the genome. This is very dramatic. This is probably the most dramatic image that we have. So can we detect any of these epigenetic changes in accessibility in vivo in humans? This is a project that I started pretty randomly with Claudio Franceschi from the University of Bologna. We wanted to understand what's in the blood. So this is a liquid biopsy from individuals, and he works with centenarians. So we started studying what kind of DNA we can identify in the blood of people of different ages. And this is what happens. We had 12 individuals, some young, 70-year-old, healthy centenarians and unhealthy centenarians. The difference between the two is that the unhealthy centenarians are bedridden and they don't perform well in cognitive tests. And we extracted the DNA that is not bound, that is not within the nucleus of any cell. So this is either free-floating or in some organelles in the plasma. And we sequenced it. This technology has already been used in many settings in the clinic. For example, maternal screening tests for Down syndrome, acute transplant rejection to detect DNA coming from apoptosis of the transplanted organ, and also in cancer diagnostic to identify potential mutation in the cell-free DNA that indicates the activation of cancer in certain tests. And as I said, most of this cell-free DNA comes from cells that die in the body, and they go through an apoptotic process. The cell bursts. It goes into these apoptotic bodies or directly into the bloodstream, and by extracting it, sequencing, we can identify what these fragments are. And the trick that we use, which is something that we didn't develop, but it was in the literature published a few years before we started the project, is that the way cells die is there is machinery within the cells that actually kills the cell by chopping up the entire genome in between where those nucleosomes are. So what happens is one of the signaling cascades in the cells that says, I have to commit suicide, the caspases activate this cutting of the DNA so that you get these small fragments. And by mapping those fragments back into the genome, we can detect where these nucleosomes originally were. So as you can understand, the more nucleosomes in a region, the more close the region, the fewer nucleosomes in the region, the more accessible it is. So we had an in vivo way of looking at accessibility without using any assay. It just comes out of the fact that when the cell goes through apoptosis, this is actually what happens. And what we saw is for young individuals, with respect to young individuals, we saw that the nucleosome signal was smoothened with age. So this is a young individuals, and this is an old individual. And as you can see, that difference in accessible and non-accessible is reduced in old, which is similar to what I showed you in terms of the senescent cells, as you can see here. That was similar to what is happening over there. And in healthy centenarians, actually, the loss is actually less dramatic. So it looks like healthy centenarians have a more robust way of maintaining the accessibility, the difference in accessibility of the different part of the genome. How am I doing on time? It's, I have 10 minutes, so great. We looked also at these transposable elements. I told you these transposable elements are actually in the regions that are supposed to be silenced. But what we noticed is that there was a loss of nucleosomes at the promoter of these elements, which indicates that with age, these might be expressed more. So some of the features that we see in senescent cells might actually be features that are in also normal cells as they age. As you can see here, L1HS is the most, is the only autonomous element that can jump in the genome. Basically, it encodes for its own transcriptional, it encodes for its own transposable machinery. Basically, it encodes for 2 proteins that allow this RNA to go back into the nucleus, get retrotranscribed into many parts of the genome. And this one is probably one of the one that gets activated as we age, and definitely in senescent cells. The last thing we did is we used the phasing of the nucleosome on top of genes to try to predict what are the genes that are on or off in the cells that die, and that is an indication of what are the tissues of origins of these cells. And this is what we found. So what we found is that in older individuals and in unhealthy centenarians, we detected the potential presence of cells that died, that have a signature in the blood, that do not come from the hematopoietic system. They are not in the blood. So we see more of other cells, which indicates that potentially there is a cell death at the level of many other tissues, especially in old individuals and unhealthy centenarians. We didn't see that much in healthy centenarians. And so that's another indication that they might be protected. So if I have another couple of minutes, I want to just show you something else that we're really excited about, and it ties, again, well with the first talk this morning. And this is, you have seen that it is important to study the 3D-dimensional structure of chromosomes to identify how these changes are linked to the change in gene expression in these cells and ultimately in the phenotype. And so in collaboration with the Wu Lab, we are trying to improve our ability to use super-resolution microscopy to walk through an entire chromosome. So this is a collaboration with the Wu Lab and Shamtanu and Guy, 2 postdocs in the lab, and a few graduate students in my lab. And this takes advantage of the super-resolution technology. We're using a Vutara 353 microscope. So the super-resolution technology allows us to visualize probes that target the DNA with a resolution that goes beyond the diffraction limit. So the nominal resolution in the XY axis is about 15 to 20 nanometers. In the Z-axis, it's a little bit bigger. And so what we do is we designed probes, and this is a pilot study, so we couldn't tile the entire the entire chromosome. This is chromosome 9. It's about 140 megabases. So we tiled sequential regions, and we tiled them in terms of TAS, what I showed you before. And we walk through the chromosome, and each time we walk, we take a snapshot of what the chromosome looked like. So what you will see is how this actually happens in the experiment. I will show you as we walk through the chromosome, as the image gets acquired and processed, and each dot that you see in the images that are going to come up is actually a single probe that targets a single location in the genome. And so this is what you have. So we start with the first tad, and it appears here. And they are color-coded by what compartment they are. This is a compartment that is a B compartment, so it's supposed to be compact and inactive. And then we continue with another B compartment. And as each one of these iterations, it's an iteration of imaging. This whole experiment took about 2 weeks of continuous acquisition, and it's about 20 terabytes of data. And so I'll just continue to show you how the acquisition worked. So it is not the full chromosome, as we said. It's still very expensive and time-consuming. We're trying to optimize the walk so that we can do it with fewer steps and with more cost-efficient way of doing it. And but this is what we have right now. You see the two chromosomes. This is homolog 1 and this is homolog 2. They're both chromosome 9. And I don't know if I can show you the movie. Yeah. So this is the structure. They are not all connected to each other. You don't see a full chromosome here because we couldn't image everything. But the idea is to be able to do this sort of study that will give you the shape of the chromosome, the density of the DNA at each location, for all the chromosomes in the nucleus. And ideally, looking at different conditions, we will be able to see how chromosomes change and how that affects gene expression. And with this, I would like to thank you, all the people that are contributing to these studies that I showed you and all the funding agencies that made this possible. And thank you. Sure.
So, excellent. I think we need to take aging now seriously. So, question. Have you done research or has anybody done research to find out up to what point can you still unfold the cells and induce pluripotent stem cells? I'm assuming that senescent cells can't be reverted back.
So senescent cells cannot be reverted back.
No. Okay. So where's the cutoff?
So, well, that's a very interesting question. I don't know if anybody has tried, because what you do is you induce senescent cells into senescence in different ways. So depending on the system you use, there's a different dynamics. Replicative senescence takes about a month or more. Oncogene-induced senescence, where you drive the damage, takes about 2 days. I don't think anybody has tried to stop in the middle and try to reverse that. And so I don't know. But usually when they go into this, when they have a sufficient amount of damage, they go into senescence. So as long as you try to revert them before they accumulate a sufficient amount of damage, it might be possible to revert it. There is one paper that shows that if you, for replicative senescent cells, if you overexpress telomerase, and you start to elongate the telomeres again, and they exit senescence. The problem with all of these studies is that they're done in culture, and the fact that cells start to grow again might also mean that there was one cell that was not fully senescent. And that cell starts to divide, and then all of this. So demonstrating that that doesn't occur is usually difficult. There's different camps in the field, but in terms of reversal of senescence, I don't think it's possible. I mean, I might be wrong. Yes?
Fascinating talk. Just a quick question. Is it possible to revert between the open and closed states of chromatin by playing with the methylation status? So, for example, could you induce a siRNA-targeted methylase?
Oh,
and
yeah, you can do that. Actually, I've seen a recent talk from Basman Celso. He's an expert in those interaction between the DNA and the lamina, which is the periphery of the nucleus. And so he has a system now in which he can actually activate a gene that is embedded in that closed region. And what he demonstrated is that the gene itself, because it gets turned on, it detaches from the periphery and it goes into almost the center of the nucleus, and it becomes exposed. But the exposure is very precise. So if you have an array of genes in that region and you only turn on one, that's the only one that comes out. And so in principle, I think it is possible to be very specific in altering the openness of certain regions. That's in the case of this possible.
Right. So one could theoretically use a methylase to close down the ALU or the retro transposon sequences and prevent senescence?
So yeah, well, so first of all, the activation of the transposon element is a late event. We think that it contributes to the inflammatory response, but it's not a trigger of senescence. Second, there are many copies. So in terms of line 1, there are about 400,000 copies of line 1. 200, I think, 1,000 are full length of those 400,000. And only 100 are active. But the fact that they express a gene is sufficient to induce inflammation. So it's hard to target all of that.
So you're saying I'm going to get old?
Sorry?
You're saying I'm going to get old regardless?
Yeah. Yeah, I mean, yeah. So we have to find a different way. We have to clear them. We have to clear them.